My lab works both on bacteria and on fly, and we study how organisms navigate chemicals. And Massimo this morning did a fantastic introduction to olfactory navigation. And so I'm not sure I'm going to be telling you anything really intelligent about data-driven or concept-driven, but I'm going to try to point out when the project was driven by data or by more conceptual aspect. This is a project that was entirely driven by a super talented postdoc, Nirag Kadakia. He had the help of another postdoc also, Mahmoud, and a collaborator with whom we could not have done that, and you're going to see why, is because he's an expert in vision, and his lab is next to mine. And so having people with different expertise next to each other is a great thing. Okay, so the question is how, as an animal, can you navigate such complicated odor plumes? And as Massimo was saying, navigating odors to their source is really important for survival, to find mates, to avoid dangers, to find food, etc. No? And the question is, what strategy can you use as an insect to do this? And one of the key difficulties is that you cannot see the odor. And so it's very difficult to image the odor field and the animal at the same time. And so a piece of data that Massimo showed this morning is that beautiful data by Kennedy et al. What he didn't mention is that together with the odor, they also released bubbles of soap bubbles. And why they did that is so that they could draw this diagram where there's thin lines and thick line. The thick line is when the moth was inside the bubble field, and the thin line is when it loses it. No? And so what we learned from this is that when the animal encounters the odors, it turns upwind. So it uses the direction of the wind to go up towards the source. When it loses it, it goes crosswind to try to reconnect with the plume. Okay? So the basic strategy is to go upwind when you find something and then go crosswind when you lose it. And more recently, there's been experiments in the lab where still, because it's hard to see the odor, one thing that you can do is that you can establish a very straight plume in a laminar wind. And then this is data from Michael Dickinson's lab where he looked at flying flies. And so since he knows where the odor is, he can tell when the fly is encountering or losing the plume. And again, he found that the fly reflectively turned upwind when they're encountering the plume. And this is data by Kathy Nagel, who is at NYU. In this case, she has walking flies inside basically little tunnels. And what she can do is you can change the odor concentration very suddenly everywhere. So there's no spatial structure, but there's a temporal structure. So either the odor is on or off. And when the odor is on, the flies move upwind. When it's off, the fly is looking around. And so basically, all of those results still point towards the fact that the fly is going to go upwind when it detects odor. And then Mahmoud in my lab wanted to see if he could look at a bit at a finer level the decision-making of the fly when they encounter these discrete packets of odor. And so he was, for the longest time, trying to find fluorescent odors that he could image, but the concentration he had to use were toxic for the flies, so they would die. And so, but then one day he was lucky, he discovered that the flies, when they're hungry, they love smoke. And so you can see smoke, and so you build this beautiful assay where now he was integrating the signal here, so this is the type of signal you see, and you get the orientation and the speed. And so from this rich data set, you can do then inferences of models of behavior and use likelihood estimation to extract the parameter of those models. And the basic results was that the flies in this type of environment in particular, they were basically doing saccadic turns at random, but they would bias those turns in the upwind direction as a function of the whiff frequency. When the frequency of whiff were higher, they would bias it upwind. Okay? So that was entirely data-driven. But so if you look from about 100 years of result, the bottom line is that the directional cue is provided by the wind, and the timing of what to do is provided by encountering with these odor packets, which tend to be discrete because of what Massimo was saying this morning. No? And so the question so looking at the data for Mahmoud, one thing we noticed is that we realized at some point, that the flies seemed to know if they were on this side or this side of the plume. And we was like, how could they know that? So we could not figure that out. And so we wonder if there was some other cue available that was not just the wind. And so one possibility is that you can measure gradients. So the flies, this is the head of a fly, those are the eyes. These are the two antennae that they use to detect odors. And this little thing here, the arista that they use to detect wind direction. And if you compare the concentration on those two, between those two appendages, you could measure gradient. And so larvae of flies, for example, have been shown to be able to navigate up smooth gradient of odor. And flying flies, when they're tethered to a pin, you can suspend a fly to a pin that is attached to a magnetic, a little magnet, so they can turn like this, no? And then you blast some air towards them, and you can odorize that air. They're going to stay in the odorized stream, and if you remove an antenna, then I have a much harder time doing it. And basically, there was a paper by Mark Fry showing that they were using the differences between the two antennae to stay inside. So they can use, apparently, the differences of the gradient. But in this type of plume, and I think Massimo said it at least 3 times, there's no gradient in this thing. And if you are far from the source, or if the signal is intermittent enough, the gradients are going to point in many directions. But if you look at this, there's still some regularity. Typically, you see the smoke coming down here and spreading away from the center line. No? And if we cannot really see with this lighting, but basically, if you were to see here, oops, I stopped it. Here you can see that there's packets passing, and they have some sort of coherence. No? There's a correlated motion of those packets. So the question or the concept was an hypothesis. Is it possible that they're actually able to detect the motion with their two antennae rather than just the gradient? No? Can you detect motion? If you can detect motion, you should be able to detect the motion of those packets and maybe use that to navigate. But in order to answer that question, the problem is that the odor and the wind are very correlated, no? Because they're transported by the wind. And so, before that, the first question we said, okay, is it possible that there is this? So let's look at our data. Can we establish a correlation between the motion of the odor and the behavior of the fly? So we took again those data where we have the trajectory of the fly and we have the antenna here. And so you have this type of data. Now you have time, walking speed, fly orientation, and then along this trajectory you can measure the concentration, the gradient, and the odor velocity, which in this case we extract by basically looking at a correlation between frames. No? And actually we look at the correlation along this direction of the antenna perpendicular to the fly. And so if the fly, so this is a function, the behavior in function of the odor concentration. So as the concentration becomes high enough, the fly is responding. And you see that if the fly was oriented in this quadrant, like in this direction, when it detects odor, it turns upwind, which is in this way, that's why it's positive. If it was oriented in this direction, it turns upwind, which is negative angular velocity. And if it was already upwind, well, it's just going to stay like that, no? And if you do the same thing for the gradient, while it's already upwind, there's no differences. But if you do that for the velocity, you see that there's a strong correlation. When the velocity is positive, which is in this direction, it's going to turn this way. If it's over here, it's going to turn this way. So there seems to be a correlation between the behavior and odor velocity. But that's just a correlation, so it doesn't really prove anything. And so how do you go about to see if really they can sense motion or not? You have to decouple the odor from the wind. And neuroscience has this new trick, which is called optogenetic. And my neighbor, Damon Clark, had been developing this system of projector to project patterns on flies because he's studying vision. And so he was looking at how the flies are responding to those patterns. No? And what you can do is that you can express channel rhodopsin. So those are like protein that respond to light and a channel that really drive a current inside the neuron when you shine light on it. And you can express that into those receptors that normally sense odor. No? And so what you can do then, you can do a virtual plume. You can have, so this is the assay where normally the flies are walking. By the way, I didn't explain before, there's 2 glass plates, and you can drive a laminar wind between, and then you can project on top this light. Or you can have like odor coming, or you can just do the light without the wind. And you can also do this type of assay where there's like very discrete packet that you can orient in the way that you want and so on. No? So now you can do an assay where there's no wind, but there's motion of odor. And so the first thing that you do is to say, let's put some bars of virtual odor and move them and see if the fly respond to it. And so as the bars are moving from right to left, you see that most flies are oriented right, and when the bars are moving from left to right, they were orienting in this way. So clearly they seem to be responding to the direction, and you can. So this is when we express this channel in all of the olfactory receptor neurons. Also, they have multiple of those neurons. But if you have a single antenna, the effect disappears, so you have no more response. But you can also use a single one of those neurons is enough to drive that behavior. And, but still, it's possible maybe that they're still responding to some sort of gradient of those edges. So what we did then is that we made those bars much wider. And we compare what happened when the on edge of the bar is arriving on the fly versus when the off edge. So when the bar arrives or when it leaves the flies. No? So if you're responding to gradient, the fly should turn left here, but it should turn right here because it's more intense on the right. But if it's responding to motion, it should turn in the same direction in both cases. Okay? And so then you can do an experiment where you conditional on the orientation of the fly, so you're passing those bars and then you analyze your data and you sort it by how the fly was oriented when the bar arrives. And so if the fly was already oriented perpendicular to the bar, there's going to be no difference between the 2 antennae, and so there should be no response. And then if it's like this, it's going to be a maximum response like in this drawing. No? And if it's gradient sensing, you should see opposite responses. If it's direction sensing, you should see the same thing. And if you look at the data, you see that basically really they respond to this motion. So now we know that they're responding to the motion. The next question is, what's the computation that they're using to responding to the motion? So this again is more conceptually driven part of the talk if you want. Turns out that in the field of fly vision and in the field of vision in general, there's been an enormous amount of study about motion detection. No? And so they have developed all sorts of stimuli to test ideas on psychophysics experiment and so on to test response of flies. And one of those, which is a classic stimuli that they use in the vision field, is this. So what do you see? You see it moving left, right, or right, left? In what direction is this moving? Who says it's moving this way? Who says it's moving this way? All right, so most of you are saying it's moving to the right, no? Which is kind of strange because this thing is actually not moving. What you have is just pure correlation. There's no net motion. So what I'm doing is that putting like a pixel, or what Nirag was doing, I'm just like there for the fun, So there is a pixel, and then the next time step, there's a high probability that the pixel on the right will be on and so on. But it's not full probability. And so you see that there's a correlation to the right. And so if the flies are using some sort of correlation-based analysis method to detect the motion, which we know in the visual system, you just experienced it. No? In this signal, there's only correlation, and you respond to it. So your visual system is actually using some sort of correlation-based analysis. Well, so the flies, they do respond to this exactly like if it was motion. But now it's cool because you can say, and in this case there was wind, but you could say, what happened if I put the correlation in the other way? So I'm going to put the motion of the odor against that of the wind. So it's going to be very confusing for the flies. They should be confused. And they are. So the data really shows that they get confused by that. Now you can do something even more interesting. So another classic set of stimuli in the field of vision is something that's called reversed phi stimuli. So here you have, is this moving right, left, or left, right? They should be different. Which one is moving to the right? This one or this one? This one, yeah. So you can detect that. But they're both correlated to the right in this case. This is correlated to the right, like I was saying before, and this is correlated to the right, but with a negative correlation. What we're doing is that we're changing the sign, the color, we're putting the opposite color on the right of the pixel. So reverse the sign, and your visual system again detects it, change its orientation because of the correlation response. You're responding to the correlation. So do the fly respond to the same thing? And they do. They basically completely reverse everything. So the flies are really responding to the odor motion illusion, so to the visual illusion. But since we have this light sensor in the olfactory system, they respond to these visual illusions. So now what's the classic zero order model of motion detection in vision is what is called the Hasenstein-Reichardt two-point correlator. And this is a model that was developed a while ago and has been a constant in the field, has been very useful to basically design experiment and test this. And so the idea is simple. You have two detectors that are separated in space, and when the signal arrives on the, so imagine there's an object moving from left to right. When the object activate this receptor, but this line has a delay. You can, for example, filter the signal with something like this, an exponential filter. But when the signal arrives here, it's not delayed. So basically, the two signals are going to be coincident here, and you're going to get a positive output. If the object was moving from right to left, the signal would go through before this one arrives because there's the delay, so you get 0. So this basically detects everything that moves from left to right. If you take two of those arranged like this, and you take the difference, now you have something that the output here will be positive when it's moving from left to right and negative when it's moving from right to left. So we say, okay, if they're using some sort of delay type correlation analysis to basically respond to the motion, it would be interesting to be able to extract that time scale. And so if you do the calculation for that, basically you expect a relationship between the delay between the two sensors and the turning bias that looks like basically an exponential with a time scale which is going to be given by the delay. And so if you take out the data, so we have tons of data, we know the distance between those two, we know when the bar arrives, we know the orientation of the fly, so we can, for every response of the fly, we can extract the horizontal axis, and we also have the turning bias, and we find that the time scale that seems to be, in this data set basically is on the order of 20 milliseconds, which actually makes sense. So it's totally within the range where the olfactory system would be able to compute this motion. So now, next question is, how do flies combine wind and odor directions? So since we can dissociate wind from odor, we can do this type of experiment. So you can flash the light everywhere at the same time. So there's no temporal difference between the two antennae, no? And so now this is the, if you want, just the response to the wind. So there's an odor, I should turn upwind, no? And the flies do turn upwind. So when the odor arrives, you get this kind of cosine response as a function of your orientation, no? But you can also do the one where I was showing before. There's no wind, but there's just motion of the bar, and you get also this. And so in this case, the wind and the motion of the bar in the same direction, and they look very similar, so we thought, well, maybe they're just going to linearly sum those directions. So if they were to do that, you just get this dashed blue line. If you look at the data, it's like right on top. So it suggests that maybe they're like summing just those two cues. So they have two directional cues, and they're summing them. Another thing that you can do is that you can put the motion of the bar perpendicular to that of the wind, and the sum would be the dashed green line here, and you see that the data again seems to be more or less aligning with it. And then you can do the reversed one where, that's not possible in reality, but basically the things are going against each other. And you see that basically they seem to cancel each other, which is also, you see in the data. So to zeroth order approximation, the flies seem to be responding to the sum of those two Qs, or like being able to use the sum of those two Qs to navigate. Okay, so now the final thing is, how is this odor motion now used to navigate? What would make sense to do? No? And so we go back to a bit what Massimo was saying this morning. So imagine you have a source of odor there, and because of the turbulence and the transport, so imagine you have wind in this direction, but it's turbulent. Those packets of odor will be basically spreading in a perpendicular direction to the mean wind direction. And you can get a very simple model of this, for example, by just assuming you have packets that are diffusing with molecular diffusion, but they're performing a random walk perpendicular, and that random walk is driven by a correlated motion of the turbulent field of velocity. And actually this was, figured out exactly 100 years ago by G.A. Taylor in this really awesome paper. And so if you take that, the effective diffusivity in this direction is much larger than the molecular diffusivity. So you get correlated motion of those packets, and they tend to basically spread away from the center line. And so if you take, on average, the mean wind direction in this case would be kind of always in this direction, but the mean Euler velocity field is pointing away from the center line. And so that's what suggests to us is that if the fly is able to somehow detect some of this motion, it has an additional cue from that of the wind that might enable help the animal to go towards the center of the plume, or track better the center of the plume. If the animal gets completely off the plume, then there's nothing you can do. You have to basically go back to this either in-foot-axis strategy or whatever strategy that you have to have some sort of exploration mode again in order to reconnect with the plume. But as long as you're inside, this should help basically keep better with the center of the plume, which can move away because even though you have turbulence, you also have like large-scale motion of the plume laterally. So then this brought us to do a few last experiments, which is let's do the following thing. So if this lateral motion is important, if we put wind in this direction and we put bars moving out, which is more natural than bar moving in, the fly should do better at navigating here than here. And so this is what they do. You look at the behavior, and what you realize is that as soon as these things start to taper here, these guys are doing okay, but these guys are not. And it's because if they go against the motion of the bar, they're going to go outwards, while like these guys are going to move inward. And if you look at the performance differences, there's like a significant differences in the number of flies that we count in these two boxes. But then you can do something even, another thing to say is that you see in the front end of this, there's a clear concentration of the fly around you see that the distribution of the fly in the vertical direction here is much more peaked in this case than in the inward case, where it stayed spread. So now, the last thing we can do actually is that you can play this. So this is the real smoke plume I was showing you before. I can play that plume straight on the fly, but I can also play it reversed in time. So when you play it reversed in time, you basically invert the correlations. And so everything else is kept the same, but the correlation have a negative sign. And so how do they do? You see that basically there's a significant difference again in performance between this case and this case. So they seem to be really using these correlations And if you mess around with it, basically you degrade the performance of the flies at navigating to the source. And so this is what I wanted to tell you. The take-home message here is that the olfactory system can detect motion of other packets. And the other motion detection seems to be obeying a correlation-based algorithm, which is similar, at least, to what is believed to happen in the visual system. And we can use basically the visual system stimuli to drive basically similar behavior, but with the olfactory system. So that's interesting because now that brings the question of, okay, there's a motion detector somewhere in the circuitry in the olfactory system, and the next step would be to go and try to discover where it is in the olfactory system. Another thing that's interesting is that olfaction is much older in evolutionary time than vision. And so the fact that there's a computation that is similar already in this system, it's an interesting thing in terms of thinking about the evolution of those computations in brains. No? And the other motion provides a directional cue complementary to that of the wind that points toward the center plume, and that's why we think it is useful. And we think we showed that the flies are exploiting this to navigate to the source. And if you want to read more about that, it is into this bioarchive paper. This is the paper where we analyze the smoke behavior, and this is another paper we just recently published where we removed the motion detection. We only flashed the arena, but with different frequencies and duration of the signal. And we're looking at what happens. Okay, so this was totally driven by Nirag Katakia, who, by the way, was a Schwartz computational neuroscience postdoc. So I don't know if you know about the Schwartz initiative, but it basically pays for theoretical computational neuroscience postdocs in different labs. And so he joined my lab. He published like 2 theoretical papers, and then he was sitting next to Mahmoud and started to play around with projectors and so on and design this whole thing. And since Damon's lab is like literally next to us, we sit next to each other, he could also talk to them, and so this is how all this happened. So